Oh, and that was cool, right? You guys A-okay on recording? Yeah, great. And then Jon Monroe from James Madison University. And I think, Crispin, you're going to kick it off, right? And so thank you, everybody, for being with us this evening. And thank you to our speakers. It's really a treat to have you guys here. And so I think I'm just going to turn it over.
Right. Well, thank you, Lee. Thank you all for being here. Thank you for those of you that are online taking part in this meeting this evening. I haven't done this kind of thing for quite a while, and I haven't often spoken about ASPB as an organization, which is what Lee invited me to do. So you'll hear a little bit about a professional society in the plant sciences before we segue into some real science with Jon talking about his research on starch and evolution of enzymes related to the metabolism of starch. I'm going to be following along on my notes screen. So if you see me looking around in strange places, it's because I'm trying to find the cursor's location on the. I wrote too many notes. All right, I'll just dive in. ASPB's mission is right here on the screen. It's quite simple and straightforward. And even as we evolve as an organization into a regular cycle of strategic planning, the mission basically remains the same. We want to promote the growth and development of plant biology as a science. We want to encourage and disseminate research in plant science, primarily through journals and conferences. And we want to engage and educate the public and policymakers about the importance of plant science and plant science research. That latter bullet becomes increasingly important as governments around the world put more or less money into the basic discovery research that drives so much innovation in the food and ag space. And food and agriculture is ultimately where the research that ASPB and its members and its community go. The society is officially 101 in 2025. It was founded in 1924 as the American Society of Plant Physiology with 104 members. The history is kind of interesting. ASPB split from the Botanical Society of America. It was originally a subdisciplinary section of plant physiologists, but they felt they needed their own society. So they started off with annual meetings, and in 1926, 2 years after the society founded, Plant Physiology, the journal, started publishing. We have a history. If you want to read the blow-by-blow account of the first 6 or 7 decades of ASPB, you can find this publication on ASPB's website. It was written by Jack Hansen, who was a former president of the organization and had access to a lot of information about the organization. It's about 270 pages. So if you want to kind of skip that or get the potted version, just last year as part of our centennial celebration, we published a sort of an update. And this isn't a rigorous history book with dates and she said, he said kind of information. It's more of a series of essays by some of the people who were involved in the past 35 years of ASPB's growth and leadership. And I'll speak a little bit more about some of the topics that are covered here in this document, but I will say that the first chapter, The Origin of the American Society of Plant Physiologists, is basically a potted version of Hansen's history. So you'll get 270 pages in a couple of dozen. As I was saying, the chapter 5 here, part 5, the history of funding for plant science research. This focuses mostly on funding in the US. And as an organization whose roots and whose membership is largely in academia, there's sort of a symbiotic relationship between government funding of basic research in plant science and how well we're doing as an organization. So this is actually a rather important topic for ASPB and has been for the entirety of our history, I'd say. Before we get into what we do as an organization, I thought I'd spend just a couple of minutes sharing a little bit about how we go about doing it. And effective leadership is central to ASPB's continued success. We know it's important to develop explicit and transparent pathways into leadership. We're in the process of doing that, and as we kind of formalize that process, we are looking to exploit ad hoc opportunities through some of the programs that we have that are deliberately engaged around nurturing future generations of plant scientists. The key decision-making body of the organization is a 7-member board of directors, 6 of whom are elected either directly by the membership or by the council, which is a larger representative body comprising the chairs of each of the governance committees, as well as representatives of our sections. And I'll tell you a little bit about the sections in a moment. The board of directors is a subset of the council, so they have lots of meetings. If each of these major committees is meeting every other month, the members of the board of directors are engaging with the staff and in each other as leadership, as members of the leadership every month. What we call the board of trustees is essentially a finance and audit committee. They, with the board of directors, work on the annual operating budgets for the organization, but they also directly oversee the investment portfolio of the society, as well as our annual audit, external audit that we have every year. The committees, you can see their names there, along with the staff of about 20 people. They carry out the good work of the society. We have a few different kinds of sections, categories of section. We have some that are affinity or discipline based. So an affinity section would be the early career plant scientists. We have a very robust community of early careerists who are active in the society and to the point that they have their own section with their own activities, mostly based around our annual plant biology conference. The primarily undergraduate institution section is another affinity section comprising mostly faculty who teach and do research at smaller institutions, not non-PhD granting institutions. And then the EEPP section, I won't try and say all of those words, is a subdisciplinary section. And we think as an organization, there's real growth opportunities to develop more of those kinds of groups that have a particular affinity around a topic of research that is important to the society, that's published in the journals, that's part of our conferences. The original sections were geographic, and you can see them there. Some of them are almost impossibly large, like the Western US section covers about 1/3 of the landmass of North America. So it's kind of hard to get people together across a geography that's broad. But on the other hand, the Mid-Atlantic section is 6 states around DC. So everybody can drive to the University of Maryland for a smaller meeting. And that's the main point of the sections, is to bring people together more locally so it's easier and less expensive to get together, to share information, to network, for early career scientists to find connections, to extend their career, to get to find a graduate program or get a postdoc if that's what they want to do. The sections are also effectively a farm team for membership. They're another way to build the membership of the society, and we pay a lot of attention for that reason in focusing on the diversity of the sections and the section leadership. Also, in terms of pathways into leadership at the national level, the sections and leadership in the sections is one powerful route in that regard. This here on the screen is our headquarters building. It's a home that was built in, I think, 3 sections and occupied before it was given to us by them by a family named Goody, who owned and ran a large commercial nursery on their property. You'd think that the connection between plants, plant scientists and nurserymen would be where and how and why this property came into ASPB's possession, but it was more of one of those fortuitous networking things where our accountant knew their accountant and they got to talking, and this family wanted to donate their property for tax purposes, and boom, it came to us. I'll circle back to the property at the end, but just to sort of preface that, it's actually been mostly empty since March of 2020. It's really a large mailbox and storage facility at the moment, as the society's staff has dispersed and we're working entirely remotely now. As you can see, since the 1920s, the society has grown considerably. We started off with around 120 members. By 1930, less than 10 years in, that was 420, and up to about 2,000. We're around 2,500 now, and that's a little worrying because our high point was 3,500 or 4,000, and like many organizations of our ilk lately, our capacity to keep interest in membership and the sort of the whole zeitgeist around membership has changed a lot, and we're still figuring out how to make that work. We try and work innovatively and develop new products and services for our members. We also recognize, even though we have the A for American in our name, we in fact serve a global audience, both members and non-members. We have an election open currently, by the way. If any of you online or here are ASPB members, please vote in the election if you haven't already. And I'm mentioning that in the context of the internationalism because we have a clause in our constitution that requires one of our at-large board members to be from outside of the US whenever we have a third or more of the membership residing outside the US, which we do. This is also why it's important to establish bilateral relationships with our sibling societies. We currently have relationships with India and Japan. We're working on Latin America and Spain. And we're also a founding member of something called the Global Plant Council, which is a global network of plant science organizations that is actively engaged with global organizations like the UN and the FAO around food science and plant science policy, including things like GMOs. Diversity and inclusion are increasingly important to the organization in ways that they probably weren't for the people that founded it. And one particular project in that regard is called Root and Shoot, which is an acronym of rooting out oppression together and sharing our outcomes transparently. This is still an NSF-funded project that's focused on diversity and inclusion. Remarkably, we have yet to be canceled. And we've come into the conclusion now as each Friday goes by and we're still not canceled that we should just press on regardless and hope for the best. This is something called a research coordination network that comprises ASPB. It's the fiscal agent. We submitted the proposal and received the funds, and half a dozen other plant science organizations. The point of the grant is to change how we are structured and how we operate and to create welcoming, inclusive environments for everybody associated with the organizations. We, ASPB, have made some strides in that direction. I like to think we have an Equity, Diversity, and Inclusion Committee whose remit was expanded recently to include an ongoing permanent association with our program committee that governs the content for the annual meeting, as well as working with our publications committee and editors-in-chief around appointments to the editorial boards of the journals. So it's a really important committee, and diversity and inclusion continues to be a vital part of what we do. We're also here to educate and engage with our own community and more broadly. Within the plant science community, I wanted to highlight these teaching tools in plant biology, which are created by plant scientists and educators and are aimed primarily at university teachers, although some of them, especially in the basic set, the fundamental set, are, I think, universally accessible. The very first one was called Why Study Plants and has images of Joseph Priestley's bell jar and the dead mouse and all of that good stuff. The more focused topics are proposed by the community and written by scientists who are practicing research in those areas. They cover a range of topics in areas like tools and techniques, physiology, plant development, and plant genetics. And they're freely available online through the Plant Cell, one of our major research journals. We also do a lot for K-12 teachers and educators. And this is just one example on the screen of a set of tools that we have identified that teach basic principles of plant biology, but also try and align those with more fundamental principles that operate across biology. We also have developed hands-on, inexpensive teaching lab experiments for teachers to do in the classroom to illustrate each of these principles. And again, there's a QR code there if any of you are interested in digging in more deeply. Another tool for engaging the community, and again, this is now the plant science community, practitioners, if you will, we call Plante. And this is a digital ecosystem for plant science. It's primarily a content hub, but it also includes opportunities for building networks and among groups within the plant sciences. It has a range of products and services that it operates, and it's also a vehicle for us to use to help train up future generations of plant scientists through this Plante Fellows program, which currently includes, as you can see, fellows from 17 countries across 6 continents. As I said earlier, the society's raison d'etre originally was in order to start publishing the plant physiology, um, which, as I said, will celebrate its centennial next year. And it remains a pillar in our publications and is, in fact, the most widely cited plant science primary research journal in the world. There are review journals that have more citations cumulatively, but plant physiology gets by on its longevity as well as its reach and impact. More recently, in the late 80s, Plant Cell came into being. This journal was launched in order to take advantage of the molecular biology revolution and the tools of molecular genetics that were beginning to be deployed widely in plant sciences. Since then, we've published a number of other textbooks and other kinds of innovative publications like the Arabidopsis book, which focused on that particular species as its growth in visibility and utility as a tool for basic plant science discoveries was accelerating. In our 2 main journals, Plant Cell and Plant Physiology, we have authors from nearly 50 countries over the last few years, and we have editorial boards with editors from 23 countries. In the same kind of way that the Plante's Fellow program exists on Plante, we have another tool for nurturing early career researchers, which we call the Assistant Features Editors program. And those people, competitively identified, are active members of the ed boards. They're mentored in writing skills, which I know will appeal to some of you, by other members of the editorial board, and they're essentially junior reviewers for the journal, and they're nurtured into becoming full-on reviewers as they conclude their 2-year terms. I mentioned advocacy before, and we're currently employing a legislative affairs consultant based here in DC, whose other clients are mostly other professional associations and universities, so nonprofits working for the public good. That staff has connections to the Congress and to the executive branch. Some of them have plant science expertise, some of them have policy expertise, and together they make a rich and powerful team. Our focus, as you can see here, is addressing policy and funding concerns. Sometimes that's an annual thing. Each year, as the appropriation cycle comes around, we're engaging with the Congress and working to elevate the visibility and importance of plant science research and funding for it. At times like this, it's a little bit more of a full court press. I've been on the Hill twice in the last month, directly meeting with committees and representatives. And we've held more than 24 virtual meetings with members of our Science Policy Committee and their elected representatives on the Hill. We also are putting out policy papers. Very early this year, we reaffirmed our commitment to diversity and inclusion, and we're currently working on a policy paper that speaks to the importance of international science and international scientific exchange in the plant sciences. As part of our centennial, we created mechanisms for really building on our legacy and building a network of donors to the organization, excuse me, who, for whatever reason, and many times it's because ASPB has been a part of their career since early on, are vested in the organization and its continued success and longevity. We've done this in a number of ways. The little test tube on the right of the screen there illustrates that over the course of about a 4 or 5 year fundraising campaign, we achieved a $3 million goal in donations. Some of that was through large donations and expected or unexpected bequests. Most of it was through relatively modest donations, and we're grateful for all of the people who've donated to the society, whether to the general fund or in support of specific programs and activities. One current fundraising mechanism is called Pioneers of ASPB, and the idea here is that mentees of an individual of some prominence in the organization or in plant science more generally would pony up a cumulative total of $5,000 to have their mentor labeled as a pioneer of ASPB. We're working on plan giving opportunities, and although the Centennial Challenge wrapped up last year, we're continuing to promote specific funds that target some of our programs, which one or two of which I'll mention in the future, right here. I've spoken a few times about the importance of early careerists and building the next generations of plant biologists. One way to do that is through a summer undergraduate research fellowship. We call it SURF, and the students who receive these are surfers. They're typically second or third year undergraduates who may be on the cusp of making a decision about whether they want to go to grad school. Some of them are pre-med students and carry on in that vein. Some of them don't end up doing biology at all. But there are a good number of surfers who are still active in the society and playing leadership roles. So it's kind of a diamond in the rough program in that regard. And it's one of our key fundraising tools right now. I mentioned Plant Day Fellows and the journal AFEs. We also make full ride travel awards to people from marginalized communities to come to our conferences every year, and a whole slew of other travel awards to just broaden the base of plant biology conference attendees. One innovation that we started after the COVID pandemic allowed us to start meeting in person again was something we call Plant Science Saturday. It's all very good to go to teacher conferences and to have passive engagement tools on the website, but we wanted to take plant biology into the communities where we were hosting our conferences. And Plant Science Saturday is the tool we've designed to do that. Our first post-COVID conference was in Portland, Oregon. And for those of you that have been there, the conference venue is away from the downtown area and really any populated centers. So that one didn't work out so well. But subsequently in Savannah and Hawaii, and again this summer in Milwaukee, Wisconsin, we've partnered with farmers markets to bring our Plant Science Saturday event into the communities that we're visiting. And this has been a real success because there are people who are interested in food and plants coming to those venues. Both in Savannah and Honolulu, it was about 50-50 local population and tourists. I think Milwaukee will be mostly people from Milwaukee, but regardless, it's a really great way to bring plant science to young people and their families. Excuse me. And we do go to conferences for teacher associations, the National Association of Biology Teachers and National Science Teacher Association, where we bring the resources we have for K-12 education directly to the teachers who are working in classrooms and hopefully helping our youth of today to understand the importance of plants. Speaking of the future, we continue to have a global impact. And as I said earlier, we want to expand that by developing formal relationships with other organizations. Our conference in Honolulu last year included attendees from 55 countries. Next year, in 2026, we're meeting jointly with our Canadian siblings in Ottawa. And when we've met in Canada before, we've seen that it provides an opportunity from people who either simply cannot get visas to travel to this country, people from Cuba, say, or Russia or Iran. They're able to attend the conference when it's held in Canada. And I think under the current circumstances, we'll probably where many of our attendees attending the meeting in US are having trouble getting visas from Nigeria, from India, from you name any other country. Um, we're hopeful that by convening in Canada, we'll be able to further extend that global footprint. Our journals are extraordinarily diverse in terms of the geography of the authors. And getting back to the point I made earlier about funding, China and India are currently investing hugely in basic agricultural and food and plant sciences because they need to feed enormous populations. And the governments get the connection between basic research and its applications in the field. And a very large fraction of our publications now are coming from China. India, I think, is still developing in terms of the quality and consistency of its research outputs, but part of the reason for establishing a relationship with the sibling society there is to grow our footprint in that country. And the outreach and public engagement activities of the society only expand as well. So our evolution as an organization is really important to us. Our business models depend on revenues raised from the journals, and that's fine as long as it's working, but discomforting when you begin to feel like too many eggs are in the same basket. So we're working hard to diversify revenue streams, to identify ways to remain relevant and resilient within our communities. And as part of that evolution, as I mentioned, we're a fully virtual workforce now. And one of the things I never expected to learn about in my career, especially not starting off with a bachelor's degree in microbiology and a PhD in genetics, was commercial real estate. But you'll never guess, we have the property on the market and we're looking to sell it. And that kind of bolus of income will provide us with a really, really important platform for the next iteration of innovation and evolution that we are planning for the organization. So I'll stop there. It's probably going to be easiest given the setup. If anybody has any questions, I can take them now before we segue to Jon's presentation. And I can't see anybody online unless I close this, I guess. Greg, this cursor is not moving on the screen.
It'd be hard to navigate. Oh, I gotta bring this over here.
There it is. All right. There you go. Okay. Any questions? I see one.
Is the Goody property the one that's selling?
Yes. It's not the Goody property anymore. We've been trying to avoid that terminology just because it's a lovely spot and it abuts a residential community. Actually, it's like a buffer between some office buildings and parking lots and a residential community. And so you can imagine that community is used to having this property literally in their backyards for ages, since before their homes were built. So there's a lot of sensitivity around that. The deed of covenant that enabled us to take over the property had a 20-year clause. You shouldn't modify or change or do anything with the property for 20 years. But that was in 1985, so we're well past that. I've learned a lot about the building, actually. I've forgotten the name of the president, but the first president to have a press secretary. The press secretary was the person that built the first wing of that building. I think it was the middle part as you were looking at that picture, based on digging around in the basement and having an archaeologist come and have a look at the outside of the building. And I think two wings were added on subsequently to create what we have now. But the real value, of course, in Montgomery County is the 4.5 acres of land. All right, there's a question online. Gene Rosenberg. Yes. We're also grateful and amazed that the NSF DEI grant has not been canceled. Please don't say anything to the NSF. We're thinking that just kind of flying under the radar. We'll submit our annual reports. We'll do what we need to do. But as of yesterday, they reimbursed us for all the expenses we incurred on the grant in April. John.
A few years ago, I recall the society being pretty scared of open access. Has that gone away?
The question, if you didn't hear it, was some years ago, ASPB was very concerned about open access, and has that concern gone away? So open access is a form of publishing whereby the authors pay for publishing the material versus the readers, often proxied by university libraries. So the short answer is no, the concern has not gone away. It has evolved. It's evolved on the subscription side of things as major publishers are entering into what they call read and publish agreements with universities and large consortia of universities and indeed entire countries, research institutions in some cases, smaller countries, maybe large geographically. Australia is one, Britain is another. So those read and publish agreements marry up the pay for reading side with the pay for publishing side in ways that mitigate the sort of the immediate threat of open access being imposed by governments. And you know, the regulatory scheme has changed, and in this country, as governments come and go, the visibility or perceived importance of this rises and falls. So the basic quid pro quo argument of pro open access advocates is the government paid for the research that you're publishing in the first place through the grants that the researchers use to do the research. Therefore, the outputs of that research should be free to the population to read and view and use. The counter argument is, okay, if you want the outputs of the research, read the grad student's lab notebook. What we're disseminating is a highly refined version of the research that the government paid for. It's peer reviewed. I know peer reviewers generally work for free, but it's peer reviewed in a structure that's expensive to maintain. And then it's copy edited and it's produced and it's made to look nice. The links are put into all the references and so on and so forth. So there's value added to the publication that is the value that the publishers aim to recoup by either asking authors to pay for access or asking readers to pay for access. Having said all of that, you know, there may be philosophical reasons to want to move to an open access publishing model. And this bolus of one thing that the board might choose to do with this bolus of cash is to use it to cover the shortfalls as one or both of the journals flips to an open access model. The challenge for ASPB in this regard is that if you switch to open access and have authors paying to publish, you have to really up the volume of papers that you're publishing. And whilst you're doing that, you have to work really hard not to diminish the value of those individual articles. They still have to be really good and good enough to be in Plant Physiology or the Plant Cell.
Crispin, I had a question for you. Thank you. That's great to learn more about ASPB and the history and where you guys are today. I'm curious what you're seeing in terms of interest in plant biology here in the United States versus other countries by younger, you know.
Lee's question was what we're seeing by way of interest in plant biology among the younger cohorts of wannabe scientists in the US. So it's really hard to judge. Whenever we do interact with young people, there's an enormous amount of enthusiasm, and the Plant Science Saturday events are case in point. But we're essentially swimming upstream. If you dive into K-12 biology curricula in this country, especially in the big states where, you know, whose curricula the textbooks are written for, there's photosynthesis, and that's about it. Maybe a little bit of evolution, but nearly everything is taught using animals. And we talk about, I won't say plant blindness, but the fact that despite the fact they're all around us, especially here, you know, for most of the people in this country and elsewhere, they're not really thinking about plants as plants. They're not thinking about where their cotton shirt came from or where the frites they have for dinner came from. It's just clothing or food. And until we can figure out how to change that, until we can figure out how to get plants into curricula, I think we're going to be swimming upstream. All right. Well, thank you all very much indeed. It's been a pleasure chatting, and I'm going to turn the podium over to Jon Monroe.
Sure. Get you set up here. Or you can sit. I think your talk is.
No. Yes.
Make sure we're still. Can you all see the screen still?
Yes.
Okay.
Yep.
So,
Oh.
that's all right.
Okay.
Yeah, that's important. I believe. Where did I put? Maybe, wait, is that yours too? Yeah, okay, that's the presenter view.
Right. All right. Well, thank you very much. Crispin actually mentioned me as being part of this. So thank you, Crispin, and thank you to you, all of you for being here. So my name is Jon Monroe, and I spent 31 years in the biology department at James Madison University, mostly teaching and doing research with undergraduates. And now I am retired, but I'm basically a postdoc again because it's so much fun and working in the chemistry department. So today I'm going to give you a 25-minute speed up view of one of the projects that we completed and published a few years ago about a very bizarre gene that as soon as I figured out what was going on, I just had to keep thinking about it and keep looking about it. It is a single gene that encodes two different proteins that have different cellular locations and different functions. And as far as I am aware, no one has ever found anything like that. So let me begin by thanking the NSF for the funding. The ASPB has helped JMU and lots of students. Most of those students on the left are undergraduates. And I'm going to highlight the work of Kanan, who was the only master's student in this group. I'm currently working in the lab of Chris Burnson at JMU, and Amanda was a postdoc in my lab who had a lot to do with what I'm going to talk about. So let me start with two inspirations that seem very different, but ended up coming together for this talk. One is Herb Wagner, who some of you know. He was a professor of mine at the University of Michigan. He was involved in early efforts to quantitatively develop phylogenetic trees. And that's how I started out as a botany major at Michigan. Then a number of years later, as a postdoc, I had the chance to meet Bunzo Makami, who is a Japanese scientist who purified, crystallized, and determined the structure of the first beta amylase. And I got to sit down at his computer and see in 3 dimensions moving this model of a protein that I'd been working on for several years. And to actually see it and make sense of it was a religious experience. All right, so I need to blow through the introduction fairly quickly because this story is really not about the enzyme, what the enzymes are doing, but that's all I've thought about for the last 40 years. So bear with me. Starch is a branched polymer of glucose. 2 different linkages. The alpha-1,4s are very, very common occasional branch points. Starch is a major part of the human diet, as we know. It's used to make ethanol. This is one of my wife Andrea's favorite brew. It's also involved in a whole lot of industrial applications. That's a piece of paper on which I drew the word starch using iodine. But for for our purposes today, it is used for energy and carbon storage in tubers, seeds, and leaves. At the end of the day, if you stain a leaf with iodine, it'll be full of starch. At the end of the night, all that starch has been broken down and used to allow the leaf to export sugar 24 hours a day, since it can't do photosynthesis at night. So starch accumulates as these granules inside chloroplasts, and you can purify them, and they are massive, huge, huge molecules that have a lot of internal structure, which is very poorly understood still, given that people have been interested in starch for 10,000 years. The structure is really not relevant to this talk, so I'm not going to go into it. Starch degradation, because it's a very complex molecule, requires lots of different enzymes. So the first enzymes are glucan water dikinases and phosphoglucan water dikinases that phosphorylate occasional sugars in the outer chains. Those phosphates repel one another, allowing the chains to become hydrated. So once they're hydrated, then beta amylases can get access to them and start chewing them up into maltose. They can't pass a phosphate group, so there are enzymes that dephosphorylate. And then, of course, there are branches that have to be removed. We are today only going to be thinking about the beta amylases. Beta amylases are starch hydrolases that, if this is a chain of alpha-1,4, beta amylase cleaves the penultimate bond, releasing maltose. And they work kind of like Pac-Man, that binds at the end and chews its way down to the first branch point. Most of you are also probably familiar with alpha amylases because animals have those. Plants have both. Alpha amylases cleave in the middle of the chain randomly. So one is an exoamylase, the alphas are endoamylases. All right, so when I started working on beta amylases as a postdoc in the last century, none of them had been purified or cloned from Arabidopsis. So I was able to purify the first one. And then after the year 2000 or so, the whole genome of Arabidopsis was sequenced. And then it becomes fairly easy to see that there are a bunch of them. So each one has a catalytic domain and a variable N-terminal domain. They are numbered here 1 through 9. The one I purified is now numbered 5. I won't go into that story because I named it number 1. And it is the shortest. It is catalytically active, and it's located in the cytosol. The cytosol is not where you find any starch, so we still don't know what that enzyme is doing, even though it's one of the most well-studied and most abundant beta amylases. There are 4 beta amylases that are catalytically active and located in plastids, and we know a lot about some of those, not very much about others. There are 2 plastidic enzymes that are not catalytically active. And we have worked on one of those that we call a pseudoenzyme, that it binds to another protein and activates it. Then there are 2 that are in the nucleus that have an additional N-terminal domain, and they function as transcription factors, and they're not catalytically active, which is not surprising since there is no starch in the nucleus. So today I'm going to talk about BAM2, which we discovered is active and has an unusual tetrameric structure and unusual sigmoidal kinetics. We still don't know what its specific function is. And I'm also going to talk about BAM7, which is one of the ones in the nucleus. And this was our work from a number of years ago, showing that the proteins are located in the nuclei. All right, so before I delve into a lot more of the biology of those 2 proteins, I need to bring everybody up to speed on how proteins get to their specific locations within cells. If this is a cartoon drawing of a plant cell, Each of those black lines is a membrane, and proteins cannot cross membranes, neither can starch. So where things are in cells is very important. Proteins contain the addresses for their ultimate location within their sequence, kind of like the address on an envelope is part of the envelope, and that's how it gets to where it's supposed to go. So if this is a chain of amino acids at the first part, the N terminus of that protein, if it is a chloroplast transit peptide, that would allow that protein to be targeted to the chloroplast. Others have nuclear localization signals that facilitate the transport of that protein into the nucleus. And if a protein has no signal, then it would stay in the cytosol. BAM2 has that chloroplast transit peptide, and BAM7 has a nuclear localization signal. Now, I have done an awful lot of amino acid sequence alignments, and I remember as a student, an undergrad and then a grad student, being just turned off by them. I didn't really understand them. I couldn't make sense of them. Now I do them all the time, and I think they're wonderful. So this is just a very short piece of two different beta amylases to illustrate that, when you align them, the program will try to make as many amino acids the same as it can. So what you see that's white on black is conserved residues, and black on white are variable residues. So you can quickly see that there are two different groups of proteins here. The top, I should have gone to the advancer. I think it would be easier since I'm trying to use the mouse. Sometimes one group is well conserved and the other group is not. Sometimes it's the reverse, and sometimes there's conservation throughout both of them. These are two different beta amylases from different species. One of the easier ways to make use of this data is to draw trees. So this is an unrooted tree of all of the beta amylase sequences from 9 eudicot genomes. And you can see that the Arabidopsis BAMs, there's pretty much one in each clade. The clades are pretty well separated. And BAM2 and BAM7 are in the same clade, even though they seem to be very, very different. So that was curious. That was worth following up on. So how different are they, given that they are more closely related to each other than they are to the others? Well, the top pink is the amino acids in a soybean beta amylase, the one that Bunzo Makami crystallized. And each of those amino acids comes into contact with the substrate. So in this case, what is illustrated are 2 molecules of maltose sitting in the active site. If you look at just those amino acids in BAM2s in a variety of different species, they're all perfectly conserved, as you would expect, because they're important for the function of that enzyme. Whereas the same amino acids in BAM7s from different species have a number of differences in them because it's not catalytically active. They don't have to be conserved, so they change without the protein losing its overall function. We knew, and I'm not going to explain the data, but the very first land plans contained something that looks like BAM2, and BAM7 came later. So when did that happen? And how did that happen? Well, BAM2 is on chromosome 4 in Arabidopsis, and it has that chloroplast transit peptide, and it's active. BAM7 is on chromosome 2, and it has this DNA binding domain at the N terminus. And within that DNA binding domain is the nuclear localization signal that takes that protein to the nucleus. It's catalytically inactive, as I mentioned. And the way this would have come about is a gene fusion of a DNA binding domain from some other chunk of DNA being fused to the N terminus of beta amylase 2 gene. Gene fusion events are not that common, but they do happen. The next question is, do all land plants have both? And this is one of those trees drawn by timetree.org. The gene fusion event probably occurred about 400 million years ago because BAM7 is only found in ferns and later. It's not found in the earlier plants. But interestingly, there were a couple of plants that did not seem to have a BAM2. One of those is corn, so we decided to look more closely at the corn BAM7 gene. So these are models of the BAM2 and BAM7 from Arabidopsis. This is the gene model for the corn BAM7. And we can see that the nuclear localization signal within this DNA binding domain matches very well with the first exon of the corn BAM7 gene. The second exon starts with that proline and continues on. And if you look just upstream from this first exon into this intron, there is an, sorry, I'm trying to use the mouse and I can't without touching the screen. There's an in-frame methionine just upstream from this second exon. And if there were two different transcriptional start sites, one up here and one within this intron, you could, in theory, make a long protein that would be targeted to the nucleus and a short version. The long form is already known to be in the nucleus. That was published a few years ago. The short form, if this chloroplast transit peptide is functional, could make a plastid enzyme like BAM2. Seemed rather off the wall, a little bizarre. But we were calling this a dual function gene. The first question we can ask is, are there any other plants in which such a gene might exist? And this is the corn gene model with its two transcriptional start sites proposed. And we found a couple of years ago, 14 different species that all have an in-frame methionine, either just upstream from the second exon, or one of them that has a methionine within the second exon. And you can then take those sequences and run them through various programs to predict whether they would be a chloroplast located protein or not. The three of them that we used are called localizer, target P, and PCLR. And if the number approaches 1, the probability that it is a real chloroplast transit peptide is very, very good, very high. And all of them seem to be predicted to be chloroplast transit peptides. All right, I already told you that BAM2 active site residues are conserved and BAM7s are not. What about these proposed dual function proteins? So there's what I showed you before, and this is a subset of the dual function BAM7s, and they all seem to have what looks like a catalytically active BAM domain. So that led us to 3 predictions, and I'm going to not spend a lot of time talking about the data, but the first prediction was that the short version of the BAM7 protein from corn should be a catalytically active tetramer if it's functioning like BAM2. So we synthesized a cDNA for just that short form, expressed it in E. coli, and this is the work of Claire Ravenberg, very talented undergraduate in my lab, who's now working on her PhD at Cornell. She purified both the Arabidopsis and the short corn proteins. She showed that they were both catalytically active with similar sigmoidal kinetics. And then she did SAX, small angle X-ray scattering, is a lower resolution way to get the protein structure without having to crystallize it. And the dots represent the data. The red line represents the model that predicts what it would look like if it is a tetramer. And so the data support that tetrameric model very closely. So yes, we have evidence that the short corn BAM7 protein is active and it's looks like a BAM2. The second prediction is that corn tissues should contain two different transcripts with different start sites that we call the L and the S versions. So to do that, we isolated messenger RNA from corn tissues, converted that RNA into cDNA, and then used PCR to amplify the N-terminal, the 5' ends of those clones, cloned them and sequenced them. And this is the work of Kanan, who's now working on his PhD at Purdue. So those are the two predicted gene models, the 5' ends of them. And so we made one gene-specific primer, and coupling that with a random primer, you can clone DNA that you only know the sequence of one primer for. And sure enough, one subset at the top of clones were all missing the first intron of the gene, and that would be consistent with a long transcript that encodes the nuclear version. Then there were a bunch that that started at where we predicted in the intron. I'm not going to talk about the bottom for the lack of time, but the third, so the answer was yes, there's two different kinds of transcripts in corn from this single gene. The third prediction is that the short form should go to the chloroplast and the long form should go to the nucleus. So we did some more cloning and fused DNA to a GFP, green fluorescence protein. So this is the corn BAM7 gene model with its two different localization signals. And we predicted the long form would go to the nucleus, the short form would go to the chloroplast. We designed primers to amplify the DNA binding domain, and other primers to amplify just the short form, and created two genes that we then transformed back into Arabidopsis that each have the green fluorescence protein downstream so that we can see it in living cells. So if you take a wild type cell, we isolated cells and then put them in the microscope. The red in the upper left is the chlorophyll signal, shining UV light on it. It fluoresces red. The Huxt stain in the upper right is a DNA stain which shows you where the nucleus is. The lower left is the green fluorescence signal, and in a wild type plant there is no green fluorescence protein, but the chlorophyll will fluoresce a little bit in the green signal. So when you merge those in the lower right, you can see where the nucleus is and where the chloroplasts are. So the second panel will be the long form, which we predicted would go to the nucleus, and I knew this was going to happen. The GFP signal, it coincides with the Huxt stain signals, telling us that the long form of that protein is being taken to the nucleus. And lastly, this was the short form. And now you can see all these green dots in the lower left panel coincide with where the chloroplasts are. So the short form does indeed go to the chloroplast, which is very exciting. Looking even more closely at that last panel, it looks like there's a strong green stain in some parts of the chloroplast, which could be starch granules, which would be pretty cool. Okay, what can we say about the evolution of BAM2 and BAM7? Did the original plant have one or two BAM2 genes? If there was just one, and the fusion event created what looks like a BAM7, it's very common that would have destroyed the chloroplast transit peptide, creating one protein that would have lost its catalytic activity due to mutations leading to the catalytically active BAM7. This would explain how BAM7 evolved, but it can't explain how BAM2 evolved because there wouldn't be any BAM2 after the fusion event. If there was a second version of BAM2 in the plant that the fusion event occurred in, it would have persisted and we would see it in all land plants, but we don't. We see some land plants that don't have that BAM2. So what's more likely is that the fusion event created a dual function BAM27, and that later became duplicated, one of which on the left would have lost its its DNA binding domain and would have become the catalytically active BAM2 that we see in many plants. The one on the right would have had a degraded BAM domain and would become the catalytically inactive BAM7. The dual function persists in some plants, which is what we saw in corn. So this is the last data slide to show you. The tree on the left is generated by time tree using just those species names. And it begins at the top with the chlorophyte algae Klebsiella, which has a BAM2 gene, but no BAM7 gene. And you can see the different colors on the way down are the major groups of land plants. The yellow genes on the right represent the BAM2s that we find in particular genomes. The blue arrows represent BAM7, and the green arrows represent the dual function genes that we found in a number of species. So what I'm going to now show is the inferred evolutionary history of these two genes from these sequenced genomes. The green dot on the left, far left, that 400 million years ago is when the fusion event took place to create the dual function, because all species below have either a BAM7 or a dual function BAM7. There were 4 different duplication events, one in the ferns, one in the gymnosperms, and 2 in the angiosperms, one of which created a tandem duplication where the BAM2 gene is immediately upstream from the BAM7 gene. So those are the red dots. There were 2 events within the angiosperms in which the BAM7 and BAM2 became separated. And that is pretty easy to explain. If that genome underwent a whole genome duplication event and then one was lost here and the other was lost here, then they're going to be separated. And interestingly, there are 3 groups that appear to have lost to BAM2, and we cannot figure out how that, in those cases, the BAM2 could come from the BAM7. And that would be the Solanaceae, they're too small for me to read, the legumes, and the cucurbids. So I'm hoping to find a function for BAM2 and then look at some of those plants to see if they can't do it. So with that, I will say thank you and be happy to answer any questions.
So were the 4 duplication events on the last slide, do you think were those related to whole genome duplication events, or they were potentially independent?
Yeah. Don't know. The whole genome duplications are pretty common.
Yeah, they're becoming increasingly more.
Yeah. Should I turn off the slideshow to allow you to see any questions?
It seemed to be the whole genome duplication involved in grasses.
Yeah.
But you find
Curious.
an earlier one in the monocots.
Right. And monocots have retained the dual function. No, many monocots have retained the dual function of M7. So interesting.
Yeah. Really elegant research. I'm very impressed.
Thank you.
In the grasses, there are multiple kinds of starch grains. Compound starch grains, simple starch grains, angular starch grains. Does that affect anything in these BAM genes?
Can you repeat the question?
Yes, sorry. The question, the grasses have unusually shaped starch granules. Would you say those are more common in the endosperm?
Yeah.
And endosperm of grasses is dead during germination. And the starch degradation occurs by some of the same processes, but many of them are different. They're secreted enzymes. So, but it's interesting, and I don't know that much about endosperm starch and the different structures of granules, but in leaves, the transitory starch that gets built every day, those starch granules tend to be a lot smaller and more flattened discs, and you don't see the diversity that you do in the different grasses.
Is there something different with the amylases going on in the starch when things are germinating?
Yeah, germination in barley is one of the best studied plant physiology stories of all time because people are so interested in making beer. The enzymes that are secreted by the albumin layer within the embryo are diffused through the dead endosperm, breaking down the starch acellularly, whereas in leaves, it's all happening inside a living cell, and the process is quite a bit different. Some aspects are similar, but there's a lot of differences. Don't think I'm seeing any questions here. Any other questions from you?
There's a switch in the grasses. They're having liquid endosperm.
Right.
You know anything about
No.
that?
No.
Yeah, people like the liquid endosperm.
I don't, is the chat window something I'm not seeing?
Oh,
There it is.
I think it's, yeah, it's okay.
All right, well, thank you very much. This has been
Thank you.
very enjoyable.
John, Crispin, thank you.